Hi again, everyone. Dr. Wiseman here, and we are going to do a video today on Chapter 12, the central nervous system, the structures and basic functions therein. Just to give us a little bit of clinical insight as to why we would want to learn and dig deeper into the central nervous system, here we have originally the original slide said right CVA, but I think this looks left to me. I think you're looking down towards the patient's feet through here. Be that as it may, let's go ahead and make it left, even though it says right in the slides. So the questions are, how would this affect a person's language skills? All right, so that's a question for you. I want you to think about that a little bit as we go through this lecture. What side of the body would experience paralysis, assuming this is the left side? And the hint question on that is, what does the word decussation mean? All right, so that's your hint question. So you should be able to reasonably answer these questions by the time we're done with this lecture today. Along those same lines, we have an MRI again looking through the patient's brain, and you can see the difference in the Alzheimer's patient's brain as opposed to a healthy control brain of someone the same age and gender and demographics. You can see there is quite an extensive reduction in brain mass, primarily in what we call the acetylcholinergic population of brain cells. Again, what does this mean in terms of brain function? You can think about what you know about Alzheimer's and apply it to the lessons that we're trying to learn in this lecture. And yet another example here, we have a functional MRI where we can see active areas of the brain. And I want you to notice the change and the difference here. This is a chronic pain syndrome patient. And you can see that there clearly is a change in where the brain is active and how that patient is responding to stimuli. Just a few fun facts for you. Again, not testable material, but if anything, this slide is meant to help you see why it's okay to geek out on the central nervous system a little bit and be in awe and imagine what the brain is capable of. Then when you're imagining, when you're thinking about things, you're actually learning. So 86 billion neurons, that's 8.6 times 10 to the 10th. All right, I'm saying that because I'm going to talk about 10 to the 14th here in a second. Almost 100 miles of axons and dendrites in an area that's only a few inches wide and maybe 7, 8 inches long. Not very much space for 100 miles of transmission line, if you will. So 10 to the 14th, that's 100 trillion. Okay, that's 100 trillion synapses. So we have many, many more thousands of connections beyond the number of neurons. That's because this is an integrating center. We are linking stimuli with response. And in some cases, we have to mitigate or adjust those responses based on memory and learning. All right, 50% of the neurons in the cerebellum alone. That's pretty amazing. And about 20% of the body's ATP energy. So if we're making about 150 to 160 pounds of ATP every day, 30 to 32 pounds of ATP do nothing but sustain the signaling capabilities of the neuron tissue. Just extraordinary. And a little dad humor for you. The brain is the most important organ, according to the brain anyway. Not untrue when we talk about physiologic function, but there is one kind of important correction here that I would make. And that is that the brain is actually a collection of organs, that the neural tissue in different areas have very discrete and different physiologic functions. So it's the most important set of organs, okay? So rather than one monolithic organ. Neurons do organize themselves into discrete circuits. All right, have you ever had that time when you've got like a brainworm song going on, like for instance, if you think about a catchy song, it kind of keeps going. Or sometimes if you're trying to make a decision, you can kind of vacillate back and forth and so forth. That's because neurons can and do organize in circuits. And there are four major ones. We're going to focus on two. The other two, may be a little beyond the scope of this course. Just have an awareness that there's more than two, but the two general ones I want to talk about are first the diverging circuit. For instance, the sympathetic pathway, the sympathetic outflow from the brain into the tissues, into the peripheral nervous system. If you have a response that is increasing diversity, that is to say, let's say you have a threat that shows up. Well, your pupils are going to dilate, your heart's going to accelerate and beat stronger, you're going to breathe faster, you're going to dry out, maybe get a dry mouth. You might have vasoconstriction that raises blood pressure, all of these things. Your liver starts mobilizing energy sources to create glucose in the blood. That's because that initial stimulus of whatever that threat was has led to a multi-tiered and multifaceted response, more of a systemic response. That's what divergent means. It's that it's spreading out across effectors and organ systems. As opposed to what we call a converging pathway tends to lead to specific types of responses. But not all of the above. For instance, and I use this in class a lot, is the idea of think of your favorite food, and if you're hungry, you may actually begin salivating. But you don't also have all the other parasympathetic functions occurring all at once. In fact, it's well understood that if you somehow could systemically activate the parasympathetic system from top to bottom in your body, would be extremely unpleasant when you think about defecation and micturition and salivation, nasal secretions, tears, all sorts of things like that. So the parasympathetic outflow from the central nervous system generally will target a given system, and that's what we call a converging circuit, if you will. Now, these others over here, I'm not really going to talk about all that much. There are some sort of, like, shall we say, a sort of reverberating. We see these in terms of basic autonomic function, like breathing. You see it occasionally in, I'm trying to think of some things, circadian rhythms, so I don't have, we're starting to move into complex behaviors. So I don't tend to study these as much as these two. These two are huge. These two are a little more specialized, and have some discrete functions, like I said, like in some cases heart rate, or the way that the heart regulates a delay situation between the atria and the ventricles. But again, that's beyond the scope of this course. So focus on diverging and converging. So the basic function of the nervous system is, again, integration. We may have a stimulus, like in the case of perhaps an image or a light beam coming in, and we have to get that information to the integration center. So we send it up the afferent neurons. These are the sensory neurons. We get into the central nervous system, which again I mentioned is a site of integration. And then we have a command out to an effect. We've already talked about this before, but we're just rehashing that idea that any type of stimulus response in any situation has to have some kind of a sensory input, a receptor that conveys information somewhere. It could be inside a single cell, or it could be across the entire organism, like in the case of the nervous system. You have that integration. What responses, if any, are needed? Is it an emotional response, like in the case of maybe you want to laugh or cry, or is it require motion or organ function and so forth? So this is where we integrate. Or does it require all of those? And then we have the response. Now it says motor output here, and that's true. That, in the case of muscles like skeletal muscles or smooth muscle in glands or cell functions, that sort of a thing. So we have an input, we have an integration, and an output. And so, just like homeostasis, and the nervous system is a part of homeostasis, anytime we have a type of an input, integration, effect, we have to have these three elements. And the nervous system typically connects the sensors with the effectors, either through neural inputs, through accessing resources in the integration center like experience, memory, knowledge, and then applying that to mitigate an appropriate response. A sort of quick analogy that I would talk about is if you've ever had a situation where you find yourself hesitating, well, when you're hesitating, it's because your brain doesn't really have any knowledge or memory or experience to know what the appropriate response to that situation is. And so it's trying really hard to come up with something, and then maybe you sort of ad lib or you do something along those lines. You finally reach some situation, but that's because synapses take time, and then leading to an effect. So just an interesting example of what we're talking about here. Here are the basic brain structures that you should be able to just generally recognize. Certainly the cerebral cortex, also known as the cerebrum. The diencephalon is a collection of structures right in here. Now, I often in my mind's eye include this structure, which is called the corpus callosum. It's a white matter structure, so it's mostly axons. But what you have in here, you have the thalamus, the hypothalamus, which is below that, and the anterior pituitary. The main one I would focus on is thalamus. Then you have the brain stem, and that's all of this structure right here. The easiest one to see here is the pons. The medulla is a little bit lower down here. And then you have the cerebellum, which is this lower structure right here. So midbrain, it's kind of a little piece there. Pons here, medulla there. So midbrain, pons, medulla, cerebellum here. The three main external features that we would like to know. First of all, you have the fissures, which are the big, deep cracks like between the hemispheres, the longitudinal fissure. And then we have this transverse fissure that basically separates the cerebral area from the cerebellum. Brainstem is hiding up, and diencephalon are all hiding up underneath there. But so it's really just the cerebrum and cerebellum that you're looking at there. Then you have sulci, which are shallower cracks. And so you see these sulci as plural, and gyri are the foldings and the things that you see of the cerebral cortex. So the big fissures, then we have the sulci, and the gyri are interspersed. Okay. And so the fissures and sulci, especially the major ones, the major sulci, are separating the different sort of functional regions we call lobes. The one I am not going to be particularly concerned about is the insula or the insular lobe. It's just kind of down inside, hard to see, hard to spot on models, so we're not going to worry about that too much. But we do have the frontal, and that's up front. We have the parietal, which kind of runs top but side. We have the lower side, that's temporal, and then we have the occipital. By the way, this is a nice picture here showing you the difference between a sulcus and a fissure. See, the fissure goes way down and may even have sulci popping off of it. A sulci is just a minor groove, if you will. One of the things I sort of struggle with when it comes to how deep do we need to go in this class is a slide like this. So we're going to try to dial it back a little bit. Probably the next slide is a little better, but let's just kind of tough this one out a little bit. You see this dark red and dark blue area. You have a somatosensory cortex, and then you have a sort of motor cortex right next to it. And those are, by and large, in charge of sort of your voluntary skeletal movement. So I would focus on that a little bit. I'm not worried about what controls your eyelids and so forth. You have Broca's area, which is just kind of just up from the front of the temporal lobe. This is very much a language area, and it's a language association area, if you will. Um, that's the big one there. What else do we have here? Wernicke's area would be good to know, at least conceptually. It's hard to see on a brain structure. Wernicke's is actually where the context, if you will. It's about you can speak. If you have a problem in Broca's area, you tend to be aphasic. But if you have a Wernicke's area issue, then you're perfectly phasic, but you're dysphasic. That is, you can talk all day, but you can't, the words don't have any context. The visual cortex is back on the occipital lobe. That's a good one. But these others, again, it's just a little bit too much, I think, to kind of get there. If there was any one more, I would just go with the here right at the top of the temporal lobe here, the auditory region. I like this picture a little bit better because but it's still got a lot going on. Again, you have words that you can use here, like this is a sulcus, and here's a sulcus here, and here's a gyrus, and here's another gyrus. So again, try to keep using words from slide to slide. Don't just go to this slide and focus on the information that's directly or immediately relevant to whatever the slide is saying. That's, I think, a problem that some people have is that they tend to just use the language of the slide and are not integrating it, if you will. So here's our somatosensory and motor area. That's the red and blue again. Here's our visual area in the occipital lobe. It's a little larger than that. The occipital's primary visual. What else do we have going on here that I think would be particularly useful? This is part of the olfactory system. Olfactory is the oldest sense that we have, and so it doesn't go right into the thalamus like the other ones do. This picture is getting after a couple of different areas. The cerebellum is involved in muscular coordination, as is the basal nuclei. The basal nuclei are the ones that help coordinate sort of fine motor function. If you have a problem in your basal nuclei, that tends to lead to a condition that we associate with Parkinson's. Parkinson's is what we call a hypokinetic disorder because you lose the ability to function in smooth ways. And it's mediated by neurotransmitter called dopamine. Cerebellum is often used for sort of reflexive motor coordination across the body. For instance, if you slip on some ice, you're going to start pinwheeling your arms or those sorts of things. Gifted athletes tend to apparently have a large, high degree of what we call coordination, if you will. I mean, it's no secret that now, granted, Dancing with the Stars is a popularity contest first and foremost. But if you look at the really good dancers, the ones that sort of win because of by technical reasons, many of them have been former NFL players because they have a high degree of coordination in their cerebellum with their motor cortex and perhaps some of the basal nuclei. So if you look at somebody that has essential tremor in a Parkinson's, and that's where the hand is quaking, that tends to be a basal nuclei situation. Whereas if somebody's just clumsy, that tends to be more cerebellar. And I don't have a whole lot to say about this slide. I'll be perfectly honest with you. It is showing how the brainstem and the cerebral cortex are connected. And again, this is not a particularly useful slide unless you are actually doing active dissection. Here is the front of the brainstem, the pons. But there are a bunch of other structures, and all of these structures have unique individual functions that are often associated with autonomic function. But I don't find a lot of useful information here, so I won't expect you to have to know any of this stuff for exam purposes, especially lab exam. I really only have a couple of points to make on this slide. The main thing is here's the cerebellum and how it's connecting into the brainstem and that sort of a thing. The other thing that I would point out, and mostly just for functional purposes, because Parkinson's is such a thing. If we were to section the midbrain, you have this black area. That's why it's called substantia nigra. It's black substance, and there's a red nucleus, and it's very, very dense in terms of blood vessels. But back to the substantia nigra. This is where the dopamine neurons are found that are involved in fine coordinated muscle contraction. Yes, your muscles contract in oppositional ways, like you contract your biceps brachii to bring your wrist towards the body. You contract your triceps brachii to move the wrist away. If you do them both, yes, you can hold your arms somewhat rigid, but eventually you're going to start to see a tremor appear, especially as those muscles begin to fatigue a little bit. And that's exactly what's going on, is that you're not relaxing muscle pairs while contracting others. And that's the basis of a lot of fine movement. And so if you think about a Parkinsonians, they have that essential tremor in their hands, and they're hypokinetic, mostly because their oppositional muscle pairs aren't relaxing and contracting in coordinated ways. Point of this slide, we've got Broca's area, which is again just above the temporal area in the frontal lobe. Wernicke's area is, basically in the temporal / parietal area on the side. Those are kind of the two there. I think that's fair. The limbic system is sort of our most primitive, or shall we say, deepest, most intimate aspects of our brain function. That includes what motivates us. When do we feel like we want to do something? When we're happy or sad or angry, et cetera, et cetera. And then also the integration of learning and memory. Now, your frontal cortex has a lot of that role, but it's more of a storage bank, if you will. How do we incorporate learning and memory is really the function of what we call the hippocampus. All right. We do have sort of data storage centers, and again, that's sort of the frontal lobe, if you will. But how do we access those and how do we bring those to bear? That's the hippocampus. So again, with these four structures, the main thing would be to get 1 or 2 word summary, so you don't have to remember all of this stuff. You want just a 1 or 2 word summary. Memory, okay? The amygdala, which is right here, this green structure, that's emotion, okay? Then we have the gyrus in here, which is pretty hard to see. I don't actually see it very well. It's probably this sort of this purple structure back here, but I'm not going to worry too much about it. And then we have the hypothalamus, which is one of the structures of the brain that does not have a blood-brain barrier. It monitors internal conditions of the body. It can sense blood pressure, and it helps control the autonomic functions of the brain. All right. The reticular activating system, the main thing is sleep and wake, or and let's start with that. If we don't have it entirely down, but problems with the reticular activation system are that people will spontaneously call it narcolepsy. You can even see this in animal models as well. Certainly focus, if you have a focus, it seems to help with that. And then sleep paralysis. So when you're dreaming at night, the reticular activation system basically disconnects your motor commands from the brain. And people that have issues with sleepwalking, or worse, where they're dreaming about something and then they begin engaging in the motor functions, truly terrifying behaviors while you're dreaming at night, while you're enacting those in motors. So, sleep and wake, focus, and helping protect the body from itself while you're in dream state sleep. Now we're going to get away from the overt structures to some ancillary structures that support the neural and glial function in the brain. Well, first and foremost, we have cerebrospinal fluid, sometimes referred to as CSF. If you suspect a skull or a high level uh, cervical fracture, you will often test, especially if there's blood flowing from the nose or the ear, for cerebrospinal fluid to see if there's been an issue there. You have a three-layer system called the meninges, the duramater being the outermost. If you want to see duramater firsthand, just look in the sclera of your eyes. That membrane, that whites of your eyes, are actually the equivalent of the duramater, the meninges. Meningitis, for instance, is inflammation of the meninges. And then you have this thing called the blood-brain barrier. Blood-brain barrier is almost as much a concept as it is an actual structure. That is to say, the blood vessels that serve the brain and the capillaries are very, very closely linked, so that anything that passes out of the blood into the cerebrospinal fluid must pass through a cell. Most of our capillaries in our body are quite leaky. We lose about 3 liters of fluid into the tissues a day, and thankfully that gets replaced. It gets recycled through our lymphatic system back into our bloodstream. So you do have an extensive arterial blood supply, but the glial cells seal up so you don't have leaky capillaries in the brain. If you have encephalitis, those are leaky capillaries, very, very dangerous condition. And then you have some connective tissue. There's not a lot to talk about there. The main things to think about when it comes to cerebrospinal fluid is that it's a little different than other fluids. It's got a slightly different chemical composition, doesn't have any blood cells or anything like that. It has to be circulated and filtered, and that's the role of the choroid plexus, along with and the choroid plexus, by the way, is a collection of some cells we called ependymal cells. So we have to circulate that CSF volume in order to keep it replenished with nutrients and have the appropriate pH and so forth. Sort of like the sinuses that we see in our skull, the brain has chambers, and we call those ventricles. And we could go really deep into the different ventricles. I don't think it's appropriate to worry about the individual ventricles at this time. They are lined by ependymal cells. These are the glial cells we talked about previously. They contain cerebrospinal fluid, again, what we talked about previously, and they serve a sort of circulatory role as far as supportive role as far as we can tell. And here we have a zoom in of the meninges. You have the duramater, which is the outside. You have this arachnoid, space, this arachnoid membrane, and then you have the pia mater, which is right up next to the neurons. And there is a sort of space here for cerebrospinal fluid to circulate between the arachnoid and the pia mater. Duramater is incredibly tough. And again, if you want to see the duramater, just look in the whites of your eyes. Blood-brain barrier, this just elaborates on what I was talking about before. Here's most capillaries. We call them continuous capillaries. You can see that there are these junctions here, and that allows for fluid to seep out of the blood plasma, not the cells, but the fluid. And that has to be returned through the lymphatics. With the astrocytes, what happens is the astrocytes create signals that then seal up those gaps, And so anything that moves out of the blood in the central nervous system has to pass through the cells and can't pass in between the cells, if you will. It has to pass through. That's what we call the blood-brain barrier. It's that it's the sealing up of blood vessels so nothing gets through without first having passed through a cell. A little bit about memory function, given that this is sort of near and dear to my personal heart when it comes to electronics. One of the things that is abundantly clear is that electronics, even when attempting to use them for benign purposes, does tend to alter the relationship between short-term and long-term memory. Short-term memory, you can hold things for a few minutes if you try, if you aren't reinforcing it. Generally limited to 7 or 8 pieces of information if you're lucky. Most people, that requires a little bit of practice. Most people generally can get about 3 or 4 under normal conditions. And if they aren't actively doing things to retain that information, it will be lost. Worse, if you switch tasks and now you're looking at a completely different data set, this loss of data is exacerbated, and that's the problem with cell phones, and that's the problem with social media. As far as we know, long-term memory has apparently limitless capacity. No one has ever exceeded, as far as we know, the capacity for long-term memory, and people live to be 100 plus years old. So far, so good. The relationship between short-term and long-term memory is that we can enhance that connection when we have things that have additional factors like emotional connection or rehearsal, repetition, or we are making connections. That is to say, oh, I didn't realize those two things were related. That's the point of studying in a diverse way. That is the point of studying in an active way. And that is the point of studying in a repetitive way. So please do use this slide and respect the physiology of memory. Okay. All the electronics in the world will not help you if you are not working through the fundamental architecture and physiology. So bringing this sort of all together, now we can look at different kinds of memory based on the functions of the brain's areas that we've talked about. As I've mentioned, the prefrontal cortex, we see it here in declarative memory and procedural memory. That's because this is a major data storage area for the brain. So your Jeopardy champions tend to have a high degree of connectivity and functionality between the hippocampus and the prefrontal cortex. Like for instance, my wife can recognize automobiles left and right. She grew up in an automobile family, and she knows, oh yeah, there's that 76 whatever, and I'm like, oh yeah, that's the thing with 4 wheels on it. All right, she has that connection there. Probably as much with an emotional thing with her family as anything. Then you have procedural memory, like skill sets, like how to throw a baseball or how to play a piano or how to type. Again, prefrontal cortex, only this time we're looking at a motor coordination area, the cerebellum. Walking, riding a bike, large muscle group motor memory, that's the cerebellum. All right. And then we have the limbic system, primarily the amygdala, emotional memory. And there's a lot of connection between the autonomic system here and the limbic system. So again, just try to get these things down a little bit. Again, major data center, motor coordination, emotion, and mood, and so forth. Part of why we dream and part of why a diverse set of study skills is more powerful than passive learning is that if we want to strengthen the connections of the hippocampus and the prefrontal cortex, we need to have this sort of consolidation, if you will. And what do we mean by that? We are bringing things more tightly together. That's what consolidation means. We're bringing them together. More connections, more routes to established patterns. So the hippocampus, some thalamus, prefrontal cortex are all involved in that. And dreaming is a part of this. Rehearsal is a part of this as well. And this is kind of a cute dad humor kind of a thing. I think we may have found the cause of your sudden memory loss. Actually, if that were the case, I would think that he would have motor perception and motor command issues. I think the tack would have to be more over here in the front, unless he happened to penetrate the hippocampus down here or something along those lines. But the nice point about this is that the brain is a collection of organs that have generally discrete and separate functions, and they work by talking to each other. So we have now the spinal cord, and the spinal cord is down below the brain stem, and we have the cranial nerves, and we have the spinal nerves. Now, in cranial nerves, you have to learn about sensory, motor, and mixed, right? Well, the spinal nerves are all mixed. Right. And the neuron and the nerves that emerge out of the spinal cord coincide with the vertebra with that from which they arise. So you do have these fossa that allow for the nerves to come out and span out into the appendages or into the abdominal and thoracic cavities, pelvic area, lower extremities, etc. The spinal cord is encased by meninges, so you can have spinal meningitis or you can have cerebral meningitis, and it's identical to what we see in the brain. But again, the spinal nerves are related to the individual vertebrae that are there, like C1 through 8. We've got a dozen thoracic ones and 5 lumbar, 5 sacral, and a coccygeal one. They do have a ventral emergence. That's what's shown here in the front. That's the ventral. That's the motor side. And then you have the dorsal side, which is the sensory side. That's shown right here. The sensory side, you have this little protuberance we call the dorsal root ganglion. And this one I'm gonna emphasize. All right. This is an easy landmark. It's a little bulging on the root just outside of the spinal cord. You can spot it from a mile away if you know what you're looking for. The motor neurons are the ones that command the outflowing. The efferent do not have that because the cell bodies are inside the central nervous system. But the afferent, the ones that are coming in, the cell bodies for those neurons are found in that dorsal root ganglion. And for once, we have a nice, simple, relatively simple slide here. We were showing a cut across of the spinal cord. You can find this at almost every level of the spinal cord. It does differ from region to region, but it's very, very similar across them. Now, there's the dorsal root ganglion here at 13. I'd kind of slide it a little more up here. It's a little more up in this area. So be it. This is the motor neurons coming out of here, coming out of the what's called the ventral root. Gray matter, this is where you have cell bodies, all right, nuclei. White matter is almost exclusively myelinated axons. So not a lot of, it's mostly signal conduction in those areas where you have integration in these areas. And a little more dad humor here. The human brain is amazing. It works 24 hours a day and only stops when we're taking exams. And yes, it does feel like that. I would argue that it doesn't stop functioning. It's just that we haven't prepared it properly. When we stop functioning on the day-to-day level, that's where we get set up. There's an old joke, an old saw. In order to pull a rabbit out of your hat, you have to first put the rabbit in the hat. And if your brain is the hat, you have to work on getting that rabbit in there first if you want to pull it out during an exam. So the reason the doesn't stop when we take exams is that we haven't done enough to put the rabbit in. That's the day-to-day, the small things, the diversification of the information rather than just trying to memorize. Most of these next slides are just review a little bit. What are the different lobes? Again, insula is going to be really hard to find in an anatomical way. We do have some functional things, like for instance the basal ganglia, basal nuclei. That's involved in motor functions in the substantia nigra, etc. That's really all this slide is showing you. Let's talk about a couple of structures here I hadn't mentioned. The thalamus. Thalamus here, what happens is as signals come into the brain, immediate and urgent balance and equilibrium, like walking, running, motor things like that, will route into the cerebellum. Like for instance, if you slip on some ice, like it's cold outside and you slip, you start windmilling your arms before you fall down, or you brace for impact and injure your arm that way, that's cerebellum. The other one that doesn't route through the thalamus is the olfactory system. It kind of goes into the limbic system more than the thalamus. Everything else, watch my red dot here. It goes up into the brainstem through the thalamus and then routes to the appropriate region. For instance, if it's a visual thing, like you're looking at this red dot right now, it would go to the occipital region. If you're hearing my voice, it goes to the auditory processing syndrome. If it's touch and so forth, it would be the somatosensory cortex, et cetera, et cetera. So the thalamus is sort of a network operation center. And if we have thalamic issues, you might perceive a sound as a vision, or other types of, shall we say, allodynias and hallucinations and things. The hypothalamus is really integrating the autonomic system and the endocrine system with the brain. So it's the origin of many of the endocrine signals that our body uses. It also is a monitoring center. It monitors body temperature and blood pressure and oxygen levels and so forth. And here's the limbic system. We've already talked about that. Memory, emotion, sort of your deep, more primordial sensations that you have with your thoughts. Midbrain is really coordination. This is where we find the substantia nigra for dopaminergic neurons. Yes, you got a little reflex stuff going on. So it's mostly a connection center. Pons, this is involving connection to the cerebellum. Medulla oblongata, another connection center. Reticular formation, we already talked about that. That's wakefulness, focus, and it fundamentally disconnects the motor cortex from command functions during deep sleep. And here's the cerebellum again for you. Again, posture, balance, general large motor pathway coordination. This slide here is showing the relationship between most of the cranial nerves, not all of them, but most of them. You don't have the olfactory shown in here, at least that I can see. I am missing it. But they rank the cranial nerves 1 through 12, and then the next most important piece of information is it solely sensory, like for instance olfactory? Is it motor, or like for instance the oculomotor nerve, or is it mixed? That is to say, does it have both sensory and motor function? All of your spinal nerves are mixed, and the closer and closer you get to 12, the more commonly it will be of mixed. So olfactory is exclusively sensory. Optic is exclusively sensory, but the ocular motor is motor. So you're going to have some specialization up top, but as you get closer and closer to 12, it becomes more mixed. We're going to deal with this a little bit next week, and so that's where I'm going to leave it right now. And so that's it for today. I hope you have gained something from this lecture. And we will pick up with spinal nerves and cranial nerves, peripheral nervous system next time. Thank you for your time, and I will hopefully see you soon.